Hello, my name is Jason Linville. This is CJ352, Never to Be Watched Again. And this is the mini lecture because that's what online educators recommend doing. Don't make these lectures too long, comma, Dr. in parentheses, Dr. Linville in their guidelines. Okay, so now we're going to talk about description of STR. So in this little nugget video that you're about to watch or participate live, it does, no, you guys are not recorded. Well, we're actually going to find out. I believe that the webcam it records is just me. If one of you were to start speaking, Becca, then I think your face would flash. So if anybody wants glory by being, enshrined in these recorded lectures for the history of time, just turn on your microphone and shout Baba Booey, and then your face will flash during the, I don't know what. Okay. So in this mini lecture, we're going to talk about these short tandem repeats, talk about the 13 that the FBI chose. So we're going to define some terms: locus, allele, loci is plural for locus. So just know the definitions. If I give you definitions and I say, what is this? And then you can say locus, or if I say define a locus, then you can write a short answer on quizzes and exams. Explain why alleles come in pairs in humans. So this question on the quiz or exam may come in many forms, but I think we already covered a lot of these. So some of this is review as far as you get one allele from your father and one from your mother. That's what I'm looking for. How STR alleles are described. That's sort of code for recognizing that we're not sequencing these alleles. We're actually counting the number of repeats. Okay, explain why we use 13. Well, if we just use one, that's not going to be good enough for identification. And also, we're going to introduce this that we've never talked about before, the 4 base pair nucleotide repeat. And I'm going to tell you a little bit why we don't use 5 or 6 and why we don't use 2 or 3. Okay. So let's start. Remember, what we're going for here is starting with a blood sample, converting it into a DNA profile. How do we describe that DNA profile? 13 pairs of numbers. And here there's actually 15 plus the amelogenin for biological sex determination. And these are the terms that I want you to be familiar with, okay? The locus is the location, the allele is the description of DNA. I always like to remind students who maybe have had the term allele before in high school biology, there it was dominant allele, recessive allele. But still, you were describing the DNA in the pea pod plant at that time. Big A meant dominant allele. You were describing the DNA. Little a meant recessive allele. Okay, you were describing the DNA. But here, we're not dealing with dominant and recessive genes here. What we're describing is the number of repeats. So here, the number of repeats is going to be the allele. Okay, so the terminology here, like if we look at that D5S818, that little thing that is up there, where's my mouse go? If we look right here, and this is my DNA, okay, then how we would say this is we would say, okay, for Jason Linville at D5S818, the locus, Jason Linville is an 11 and 13. We're describing me. We're describing the DNA. We're describing the alleles at this location. This is the description, 11, 13. And we, as trained forensic scientists after this lecture, know that that 11 means the STR is repeated 11 times on one allele, and on one chromosome, and repeated 13 times on another chromosome. Each person has 2 copies of each chromosome. We can see it there in the middle. If we opened up one cell, this is what it would look like. Okay, so if we look at a location on chromosome number 5, all right, right there, or as it shows right here, you're going to have 2 alleles. Okay, so every cell will have 1 allele from their father and 1 allele from their mother. So this could be the 11 and this could be the 13. And we talked about this in class, but it's worth mentioning again. We have no idea when forensic scientists do the analysis whether the 11 came from the mother or the 13 came from the father. It doesn't matter for forensic scientists. It's just, there's an 11 and 13 for this person, a pair of numbers. Okay, so if we zoomed in on these green areas here, okay, this is what we would see on chromosome number 1. Okay, and then this is what we would see on the other chromosome. Okay, now Obviously, in a sample, you have hundreds of cells. So you have hundreds of chromosome number 1, you have hundreds of chromosome number 2. But in amplification, as we're going to find out, we just amplify this little region. So at the end, when we have 10 million copies, it doesn't matter if we've started with 100 or 300 or 3,000. At the end, we're just going to have a bunch of copies of this, and the amount of chromosomes in the tube at the beginning becomes sort of irrelevant. Okay, so each person has 2 alleles. And as it says there, most of the time people will not have the same alleles, but they can. So how we would describe these people is person 1 at, let's say that this is still D5 S818. At D5, person 1 is a 4-6. That's their pair of numbers. At D5, person 2 is a 6-7. At D5, person 3 is a 5-5. You would actually say it twice because it always comes in a pair of numbers. And if you notice on that sheet with the DNA profile, it's written twice. Now, when you're actually doing the analysis and looking at the electropherograms, which we're going to get to in a couple of weeks, you're just going to see one peak. But you know that your person is not a mutant, you know, with a father and no mother. You know that it means that their father had an 8 and their mother had an 8 at TPOX, okay? So you could very easily be homozygous and have the same allele given to you from your mother and your father. It's just that typically when you're just throwing numbers out there, you know, they tend to not be the same. Okay. So what were the 13? You guys don't need to memorize these loci. What I want you to notice that we're going to talk about next is that for all of these loci, they're all 4 base pair repeats, okay? And they're different sequences, okay? I want you to know the sequence doesn't matter. It doesn't have to be AGCT. It doesn't have to be where all 4 letters have to be different, or there can't be more than, you know, 2 letters in it or something like that. As long as it's 4 letters and it's repeated back to back to back, that's fine. So I want you guys to know that, okay? What you don't have to know, but I want you to observe right now is, okay, it is somewhere between 6 and 20 is for a lot of them, but there are a couple oddballs here that go up 15 to 30, 24 to 38 that tend to have a little bit higher allele. So again, it depends on the STR that's chosen, okay, but that's sort of the range of repeats, the range of number. Notice that it doesn't go from 6 to 40, okay? So it's not like at every single loci, you're going to have some random number from 6 to 40. Oftentimes there's, you know, a selection of the alleles that could be present. All right. The other thing to observe, but you don't have to know, is over here in column 1, the STR marker. Sometimes you have that D5 number. That means it's just in sort of the extragenic DNA, sort of between genes or something. But you also notice sometimes they have a weird name like this. Why does this have a weird name? The reason that some STRs have that weird name is it refers to the gene that the STR is located in. Wait a minute, Dr. Linville, you said that these were non-coding DNA, they weren't in genes. Yes, but if you remember from biology 1 or 2, or really probably any decent party you've been to where they're talking about the human genome, you remember that within genes we have exons and introns, and before the gene, after transcription but before translation, sometimes part of that gene is thrown out in the messenger RNA, so it's never coded. STRs can be in those introns that get cut out because they never end up coding for a protein. Okay, and the reason why there's a lot of them in here is because when the FBI was picking their STRs, they were picking STRs that had been studied and that people knew a lot about. So therefore, if a STR was in a gene, it may have led to it being studied a little bit more than some other ones. But again, you guys don't need to know that. The other thing to observe, before I answer Daphne's question, the other thing to observe, but you don't have to know, is that all of these are on different chromosomes, with the exception to on chromosome number 5. They tend to be very far apart on chromosome number 5. And to do the statistics that we're going to talk about in the next couple mini lectures, the alleles need to be inherited independently. What the heck does that mean, Dr. Linville? It means you can't have 2 STRs right next to each other, because if you know, if I have an 8 on TPOX, it means that I'm 90% likely to have a 7 on CSF. it messes up the statistics. So the FBI also picked STRs that were on different chromosomes as well. Okay, so Daphne asks, does everyone have the same order of base pairs in the location study, or is it different for everyone? That's a very good question. So in some of these, like for TPOXX, everybody has AATG, okay? And it's AATG, AATG. It's just a different number. Hey, Kiki, can you go ahead and keep that door closed, please, buddy? I forgot to lock it. Okay, I'm almost done. I'll talk to you in a little bit. It's not working. All right, let me finish this up. There just might be somebody standing behind me. So in other words, where was I at here? The repeat units, the chromosomes, they need to be inherited independently. That's basically the important thing. Oh, no, does everybody have the same order? Okay, so AATG, it does have the same order. But down here with these stars, it means that they can be more complex. The thing is, if I have a TCTA, TCTA, TCAA, I'm only measuring how long that STR is. So therefore the sequence doesn't really matter. Okay. If different people have different sequences, or if my repeats have different sequences in them, it's not going to matter. We only care about how long they are. Does that answer your question, Daphne? All right. So we have these 13 loci in the genome. So the FBI picks these 13, and this just sort of shows how they're all sort of spread out, and on different chromosomes. All right. Okay, the next thing that we want to go over in this mini lecture is understanding why there are 4 base pair repeats, good, as opposed to using 2 or 3 or 5 or 6. Okay. Now, what you do not need to know from this slide is the definitions of down here, let me find it, the simple, compound, and complex repeats. Okay. And that's what we were just talking about, what Daphne's question was about. It doesn't have to be the same sequence of letters that's repeated. It can be where there's different repeats, or it can be where like the sequence sort of changes throughout the repeat. All we care about is the number of letters that is repeated and how many times, because when it comes to measuring these STRs, we're essentially measuring how long they are. Okay, so this slide I just wanted to put in to comment on that, but you guys don't have to know any of that for a quiz or exam. Okay, and this shows what we were actually just talking about, the question that Daphne asked. All right, why not 5 or 6? So we're going to do this in 2 things. We know that we're going to use 4. Okay, I want you to know that number 4. I want you to know tetranucleotide repeat. Okay, tetra is usually 4. Okay, but why not 5 or 6? I'm not going to ask you to know the difference between 5 and 6 and 2 or 3, but I want you to know the potential problems when we stray away from 4. Okay, so specifically for the larger repeat units, the problem here is that they can get very big. Okay, obviously if you repeat a 6 nucleotide repeat 20 times versus a 4 nucleotide repeat 20 times, the 6 is much bigger. And it's not as though we can't amplify and analyze a larger piece of DNA, but larger pieces of DNA or segments of DNA that we're amplifying are more prone to trouble when there's degradation. Okay, the bigger of an area of DNA, it's more likely that there's going to be some type of break in it when you just have degradation over a long period of time. Okay, or in a sample that's been exposed to the elements. Okay. The other thing is that difficult to multiplex, we need to amplify and manipulate where our primers are. We're going to talk about this in the next mini lecture. We need to sort of manipulate where our primers are, manipulate the sizes so we can analyze more of these STR regions at one time. And it's hard to visualize right now, but it's just more difficult to do when you have bigger repeat units. Okay. So 4 tends to just be easier to amplify. You have as many problems with degradation, and you can multiplex. You can do more 4s loci in one reaction than you can if you had 5 or 6s. Okay, 2 or 3s would work very well because they're very small. But the problem with 2 or 3s, and again, this is something that we're not going to go into a lot of detail about, is when we start looking at the electropherograms, like this person is an 812, okay? So we would read this as a forensic scientist going, oh, there's this peak indicates that there's an 8 allele present, and this peak indicates that there's a 12 allele present. But also on the baseline of the electropherogram, which is the sort of scientific chart that the machine spits out, we're going to notice that there's a little bump before both of these peaks, okay? And when you're using tetranucleotide repeats, that bump is small and the instrumentation can ignore it easily, okay? In di and trinucleotide repeats, these bumps get a little bit bigger and they can cause confusion as to, number one, what the actual profile is, and number two, if you have a mixture of two people's DNA, it just makes it a huge headache. Okay, so 2 or 3 nucleotide repeats, they have this problem with stutter, is what these little peaks are called, stutter peaks, and it makes the forensic scientist's job a little bit more difficult. Okay, so just know that, you know, subject to degradation, hard to multiplex, and stutter, those are all 3 of the issues for why we use tetranucleotide repeats. Okay, but I don't think the questions I'm necessarily going to make you associate the stutter with the smaller ones and the other with the bigger ones. I'm just going to sort of ask you, why do we use tetranucleotide repeats? Okay. All right. There's a mistake here. Again, we're going to stop this for discussion of STRs, and we're going to immediately start our last mini discussion for today. So let me stop recording. Thank you for listening.